Welcome back. We are still discussing optogenetic technology, and today we will try to look into how to control the brain with light. So, welcome to lecture number 42, module number 9, principles of optogenetic technology, and let us see if light could be utilized to control certain section of the brain. So, right. I think it was in 2005 or 7, please correct me if I am wrong, that at Stanford, Carl Deseroth and group of his students tried to did a revolutionary experiment that resulted in coining the term optogenetics. So, what they did is, there are certain types of algae and other microbes which depend on their survival on certain specific proteins. Those proteins are called, a group of proteins are called opsins. What are the specific peculiarity of these opsins are that these proteins, these specific proteins, upon excitement by a specific wavelength of light, will result in flow of a specific electrical current, right. There is a group of protein, opsin group of proteins present in certain algae that are photosensitive, and that result in somewhat, very crudely speaking, photoelectric effect, i.e., you send light at them of particular frequencies, particular energy, particular wavelength, and they result in conduction of current through them. Photoelectric effect naturally existing in proteins existing in certain algae and other microbes, i.e., biocompatible. When illuminated, these protein channel regulate the flow of electrically charged ion across membranes, which allows the cells to extract energy from their environment. Opsins of different types can vary in their light sensitivity and behavior. The opsin genes that make these proteins are the foundation for optogenetic technology that neuroscientists are now using to control the activity pattern in targeted neurons. So, this is an example of the various types of opsin proteins. That is channel rhodopsin, CHR2, then VCHR2 is another type of channel rhodopsin, then there is halorhodopsin. So, these are different types of proteins. If CHR2 channel rhodopsin is excited by a wavelength of 470 nanometer around indigo or blue, this allows positive sodium ion to pass in response to blue light. Sodium, potassium, all those things, chlorine are available in the body anyways. Yeah. So, if this particular protein present in a cell is excited by 470 nanometer, it allows a positive sodium ion Na plus to pass through it. If the light is not excited, this particular protein will not allow sodium ion to pass through. VCHR1, another form of channel rhodopsin, responds to some wavelengths of green and yellow light. CHR2 channel rhodopsin is 470 nanometer, another category. VCHR1, the input has to be green or yellow light, and it will respond to some wavelength, it will allow sodium ion to pass through. Halorhodopsin, on the other hand, gets excited by 589 nanometer yellow or orange light, and it allows negative chloride ions to pass through. Now, remember, these ions, these charge carriers, these electrical current charge carriers will only be possible when you have excited with this specific wavelengths only. Photoelectric effect in proteins. Photoelectric effect in proteins. Only upon excitement by a particular wavelength of light, a particular current will pass through it. So, you have relative response to light versus wavelength. So, basically this is current versus wavelength. The different types of electric current that is being sent when excited by specific wavelength of light is actually shown. So, NPHR halorhodopsin will only be exciting or only be passing current when it is around 580, 590 nanometer radiation is present. Any other radiation, either it is very less or none at all. Similarly, channel rhodopsin of different types of proteins allows the passage or blockage of different types of ions, thereby either allow or disallow a particular electrical impulse, a particular electrical current, a particular electrical pulse to pass through it. Right? Ions carry charges, so they are charge carriers and they can be used to conduct electricity basically. So, what Professor Carl Dresserot's group did was something incredible, right. So, they isolated first the gene from this algae or microbe that produces this protein. What is a gene? We have discussed this, right. If you have forgotten what gene is, go back to the previous lectures. So, Carl Dresserot team isolated a particular gene which produces these photosensitive proteins that has a capacity to produce photoelectric effect, right. It was connected with a promoter. Promoter is the beginning of a gene, the start code UAA. Remember the start code upon which the gene become functional, the start code which the person receives, from which the message become functional. It is connected with the promoter, which is then put inside a virus. This virus is then made to infect the brain of a mice. This promoter and opsin gene then connected to specific neurons. You can specify neurons by connecting it with a specific promoter, the specific promoter attach with specific part of the gene. So, you know how viruses work, right? Viruses affect the nucleus of the cell. So, this particular virus was made to attack the nucleus of the neurons present into the mice. So, scientists combine an opsin gene with an element called a promoter that will cause the gene to be active only in specific types of cell. So, this promoter is specific to a specific neurons or specific group of neurons. The modified gene is inserted into a virus, this entire gene, this gene that has been isolated from algae is inserted into a virus, which can then be injected into the mouse's brain to infect it. Gene put inside a virus, virus put inside the mice brain. Here the virus is acting as a carrier, acting as a transporter, acting as a car or a plane which is carrying the cargo. The cargo is this gene. This is gene therapy. They did it. It's a fantastic, almost Nobel Prize winning experiments. There are few beautiful experiments of science. I would rate this experiment as near the top. Gene, you are extracted it from, what is gene again? Come on, group of DNAs that has the capacity to create a protein that can express proteins. So, set of DNAs put inside a virus, virus contains DNA or RNA, put inside a virus, this virus is made to infect the mouse's brain. The virus infects many nerve cells, but because the promoter only one type of neuron make the opsin proteins, meaning using the virus as a carrier, specific neurons of the body specific neurons of the mice's brain, is made to connect with this specific opsin producing gene, opsin producing DNA. Understand it. The virus is attacking all over the brain, but there is something called a promoter. The promoter is a specific DNA which has a specific code. The start code attaches with specific neurons only. Yes, different neurons, they will not be 100% similar DNA, 99.99% similar, there will be certain differences, otherwise you will not be able to differentiate, right. Now, you have differentiated different brains, different brain cells. A particular cell, that promoter, the particular cell which has a particular DNA, which has a particular gene, is connected with a particular promoter. Promoter is the beginning of the gene, promoter is the beginning of the start message. This start message is then connected with the opsin gene. So, certain neurons are affected, mature age, old age neurons, or just baby neurons which are just forming, or neurons with large number of dendrites, or neurons with small amount of dendrites. Only those, only specific ones are attacked. The virus infects many nerve cells, but because of the promoter, only one type of neuron make the opsin protein. Which type you decide? Old, young, specific, this neuron present in this side of the body, etc. So, the virus have now infected the nucleus of those particular neurons. Sooner or later, the virus is benign. So, either it stays or your body flushes out, but the nucleus of certain specific neurons have got the opsin gene, meaning that neuron which contains large amount of gene, one part of the gene, one of the genes present in that, you have large number of genes present in one single cell, one of the gene present in that specific neuron is producing opsin proteins along with its normal function. Yeah. Neuron containing large number of genes, each gene produces specific proteins. Of that one particular protein is your opsin protein, light sensitive protein. Then they made a small hole, a keyhole type, onto the skull, the cranium of the mouse, inserted a fiber optic cable and started illuminating with a specific wavelength of light. Fiber optic probes inserted into the animal's brain can flash light onto the brain to control specific pattern of the neuronal activity. You have this much brain with millions and billions of neuron cells, neurons. Certain neurons are actually containing opsin proteins, halorhodopsin, channelrhodopsin, anything. These opsin proteins are then excited with a specific wavelength, They in turn then conduct electricity by sending sodium ions positive or chlorine ion negatives. That result in certain specific activity of the mouse. The mouse becomes aggressive, the mouse fall asleep, mouse become hungry, the mouse tries to behave differently. You can map them using light. The virus infects many nerve cells, but because of the promoter, only one type of neuron makes the opsin protein, and the fiber optic inserted into the animal brain can flash light to the brain to control specific patterns of neural activity. You have then thereby encoded certain areas, only those which has this specific opsin, halorhodopsin or channelrhodopsin, will be affected, no other nearby. Even if the neuron is nearby, since it is not producing photoelectric photosensitive protein, it will have no effect, it will not produce any electric current. Only this which will have a responsive, which will have a gene that also produce photosensitive proteins will be affected, and that is it. That is how you measure the neuronal activity or see the behavior. So, this is the entire mouse, this is the ear and the mouth, and this is the small brain inserted through cranium. A small keyhole surgery, few micron thin is inserted, and yes, you can keep the mouse alive. We will show you in the next, I will show you in the next slides. In a small keyhole like surgery, it is possible to insert a optical fiber while keeping the mouse still alive, and you measure the neuronal activity and map it with respect to behavior, right. So, see, this is the biggest advantage of optogenetics or light based specific control of neuronal activity. If you have put a probe, an electrical stimulation, an electrode, it will be affecting, even if it is small, large number of neurons, and it will be passing same voltage to both of them or group of them, and you will be not able to distinguish between which neuron is trying to do what. With optogenetic excitation, depending on the opsin protein that the neuron is encoded with, depending on the blue light, it will only result in sodium ion. If it is yellow light, it will result in chlorine ion. Negative ion flow, positive ion flow. Direction is this, direction is that. Negative ion flowing, positive ion flowing. Sender, receiver. What is the resultant behavior? What is the resultant behavior because of the difference flow of ions, because of two different specific neurons talking among each other, resulting in what specific behavior of the animal. Would you like to do something to your brain and let us probe it by inserting an optical fiber in your head, in your skull, and try to see whether you will start singing or dancing or become afraid or become very aggressive or become hungry or become asleep if blue light is excited in you inside your brain. Do you think at certain time this would be possible in humans and primates and map different areas of the brain? At this present moment, this is very hot topic, but we are still stuck to mice. I am working on this on zebrafish, the transparent fishes. I am working on this with these kinds of neurophotonic probes. So, they tried to excite a specific area of the neuron. So, see, this is the neuron with these branches, these dendrites and axons forming. They put some amount of light and then try to see the electrical activity, and you see this 573 nanometer. This is the laser, pulse laser was given instead of continuous laser. These were pulses, and they were able to figure out the electrical measurement. Obviously, you have to have some kind of electrical probe associated with it, but that can be connected with the optical fiber, which is a passive component. Like it will measure the electric current, it will not send any electric current. So, it will just be there to measure the electric current passing from one neuron with another neuron, or overall in the periphery. It will not be sending any electric current by itself, and they were able to see that, see this 2 millisecond electrical pulses passing from one end of the neuron to another end by subjecting it to blue light, basically photoelectric effect of the brain. Electricity passes as light is being given as an input. photoelectric effect. So, please read these references. This is a fantastic new topic that has opened up. I was actually debating whether this should be included in this or not, but then I decided to double down and try to teach 3 chapters based on neurophotonics and optogenetics, because this is state of the art. There is nothing more complicated than brain, and trying to analyze brain with light is the hottest new topic, right. So, again, optogenetics, neuroscientists have long been frustrated by their inability to study how the brain works in sufficiently precise detail. Unexpectedly, a solution has emerged from the basic genetic research on microorganisms that rely on light responsive opsin proteins to survive, proteins that produces photoelectric effect that passes electric current upon excitement by specific wavelength of light. By inserting opsin genes into the cells of the brain, scientists can now use flashes of light to trigger firing by specific neurons on command. This technology, optogenetics, permits researchers to conduct extremely precise cell type targeted experiments in the brain of living, freely moving animals, which electrodes and other traditional methods do not allow. Although optogenetics is still in its infancy, it is already yielding potentially useful insights into the neuroscience underlying several psychiatric conditions. So, hopefully in 10-20 years, with your help, several of these mental health disorders, mental diseases could be categorized and characterized to a cellular or a biochemical level, as what exactly is wrong from a molecular point of view that is causing this particular disease, and thereby probably we can cure it, right. So, these are the concepts that I covered for today: measurement of neurochemical signal, controlling neuronal activities, and I hope this is not too difficult of a topic, but this will continue for the time being. Please give me the feedbacks on the comment section, and I will try to see what could be done if you find this very difficult topic. This is difficult, I understand, but is nevertheless very refreshing and very interesting. So, please go through my references. Fantastic new works are going on, and I will see you in the next class. Thank you very much.